I think there are three main points that will benefit extensively from machine learning incorporation. First one is improved disease prognostication across all the fields of hematological tumors. And this needs to be done with readily available data for any clinician worldwide, not just with information that is particularly exclusive to healthcare facilities with advanced technology. So one of the focus is on the creation of these machine learning tools that can enable an improved prediction of disease risk without the need of advanced genetic information that can be applicable anywhere rapidly. So this is one of the main focus, and we are doing this in MDS, in myelofibrosis, but also in other kinds of tumors such as multiple myeloma or AML. Then there is an interest to incorporate the genomic information into this machine learning model. So we know that there are some kind of information, prognostic information that we can't grasp from just clinical data, and then we need the genomic information. And this will probably enable more refined prognostication and maybe change the lives of a few patients groups that are likely to be of different risk than predicted with the standard clinical tools. The second main group refers to drug response prediction. So we are developing more and more drugs all the time, and these drugs act on particular pathways, particular proteins, and it's quite naive to believe that all patients will benefit equally from the different drugs. So for these, we need to put into context both the clinical profiles and also the genomic information, the activation of the different pathways in the different tumors, in order to make machine learning predictions of drug response. And indeed, we proved this is feasible in multiple myeloma last year, and we are trying to implement this kind of analysis in different clinical trials. And the third point refers to the development of new drugs. Both the synthesis of new molecules and the immunotherapy can benefit from machine learning applications. And for example, there are groups such as ours who are working on the fast synthesis of new molecules, taking into consideration protein structures, or other groups are identifying new antigens for immunotherapy, considering, for example, the machine learning predictions of the binding of these new neoantigens to the HLA epitopes. So there is a broad and accelerated development of machine learning tools in oncohematology. Probably the most readily accessible tools will be the ones that we are seeing currently in the papers, such as the one that we will present here at ASH this year. But there is a lot to come in the field of personalized medicine with AI tools.